biological macromolecules, the proteins. Proteins are an abundant organic biochemical structure that we find in every type of organism. Proteins have very diverse shapes and functions. We're going to find that some proteins can serve to regulate how cells function. Some proteins are structural, simply there for scaffolding. Some have protective functions, like many of the proteins that we find in mammalian or human immune systems. They can be used as transport devices. They can be used for biological catalyst purposes in metabolism, and even some have hazardous or deadly components that we call poisons in the form of biological toxins. So a lot of diversity of structure and function in the proteins. Somewhat similar to carbohydrates, proteins are made of building blocks that we call monomers in the form of amino acids. The amino acids have a very basic structure. There's a fundamental blueprint for every amino acid building block of a protein that starts with a central carbon in the middle. That central carbon will have one amino functional group attached. So the central carbon has a side group attached. We'll see examples of what the side group really means, and a hydrogen. So an amino group, a hydrogen atom, a carboxyl group, and a side chain. That is the basic template for every amino acid. Here's an illustration of a table that shows you the 20 fundamental building block amino acids that we find in virtually every organism. The side groups are highlighted in blue. So we can see in the upper left-hand corner, the simplest amino acid in terms of structure is glycine. The side group, or the R group on glycine, is simply another hydrogen. If you take a look in the bottom right-hand corner for tryptophan, notice that it has, as part of its structure, these two geometric, I call it stop sign in a house. It's a very large, bulky side group. So amino acids have a wide variety of shapes and side groups. Acids together, we're going to use a specialized name for the covalent bond that links individual monomers of amino acids together to form what we call peptides. And we call that covalent linkage a peptide bond. Notice that the carboxyl group on one monomer amino acid is going to be linked through a peptide bond to the amino group on the next or second amino acid. And that peptide bond is not a carbon-to-carbon bond that we usually see in almost all other kinds of biochemicals, but is instead a carbon-to-nitrogen atom bond. That carbon-to-nitrogen peptide bond is actually a fairly tight covalent linkage. It's a little bit stronger and a little bit tighter than a carbon-carbon single covalent bond. In fact, sometimes biochemists describe the peptide bond as a bond and a half. It's stronger than a single covalent bond, carbon to carbon, but it's not quite a double bond that we might see in some structures. But it's a very strong covalent linkage forming the peptide bonds, which is part of why proteins make a really good source of food, good source of energy. We talk about protein being a high energy food. It takes a lot of energy in our metabolism to break up these peptide bonds. So these are really strong, powerful chemical bonds. What's the difference between a peptide, polypeptide, and a protein? It's simply the size. If we have two amino acids linked together, we call that a dipeptide. If we have three amino acids linked together, again, these are all linked together by peptide bonds, we call it a tripeptide. If it's more than two, but perhaps less than 100, then we call that a polypeptide. And then very large structures would be called proteins. So they're all part of the same family of chemicals that are formed with the use of peptide bonds. If we take a look at a protein, oftentimes we have other structures that are associated with the proteins that we call prosthetic groups. The prosthetic groups are not made out of amino acids, but instead made out of something totally different. So it has a unique structure and function when we look at whole proteins. We have the hierarchy of shapes when we're talking about proteins. Proteins have 4 levels of arrangement or structure: primary structure, secondary, tertiary, and quaternary. For the primary structure, think of beads on a string, just like a pearl necklace. The individual beads would be amino acids. The sequence of which amino acid is used in every particular position on the beads of a string, we'll talk about that later. But just simply know right now that if we're looking at the primary structure of protein, we're simply looking at the linear arrangement of amino acids one after another. And here's an illustration of a protein hormone that's called insulin. Notice that you can see that insulin actually is a very small hormone in terms of structure, made up of a few dozen amino acids, and we can see the primary structure very clearly in these 2 strands of amino acids. When we have longer strands of amino acids that are held together by hydrogen bonds, we wind up with secondary structures. That's the next level of organization. So the beads in the string of the primary structure start to fold and twist. When they fold and twist, hydrogen bonds are formed, and we wind up with what are called either alpha helices, which is a coiled structure, or a folded structure called a beta pleated sheet. So alpha helices and beta pleated sheets are examples of secondary folded structures of chains of amino acids. If we keep folding and arranging that primary structure and even secondary structures of the protein, it keeps folding and twisting and has other interactions between the side groups, the R side groups of amino acids, then we wind up with what we call polar interactions, which are described as hydrophilic or hydrophobic, water-loving, water-fearing. So we have polar interactions, we have ionic bonds that can form, and even bonds between sulfhydryl groups on distant, separate amino acids that are, if you look at the primary structure, they're just very distant from one another. But when you start to fold the primary structure around into this more three-dimensional shape, the sulfhydryls on the 2 separate amino acids get close enough, and with the proper types of biological catalysts, form a disulfide linkage, again helping to stabilize this fold or this twist in the tertiary structure of this protein. It's the disulfide linkage that we mentioned briefly when we talked about curly hair, when we described sulfhydryl groups. The reason that curly hair is curly is there are more sulfhydryl groups on the amino acids in the hair protein, and there are more disulfide linkages in that strand of protein that's forming that hair. Then you get the curly, natural curly appearance of that hair. The 4th level of organization for a protein is the quaternary structure. And so here's an example of going from the primary structure of beads on a string of amino acids, the secondary alpha and beta secondary structures, the tertiary protein structure, which for some proteins that's as far as it gets, myoglobin, which is the muscle protein that captures oxygen, that's an example of a tertiary structured protein that's complete in its shape. And here we have an example of a protein like hemoglobin that would have a quaternary structure.